Welcome, dear listener, to this webinar, Tackle the High-Hanging Fruits, Solution for Today's Challenging Drug Discovery Pipelines. Now, this first figure will be familiar to many of you working either in the pharmaceutical industry or the biotech industry. It shows how the spending to develop a new drug has significantly increased over the last decades and has now reached a point where it can easily cost up to $1 billion to develop a new drug. Now, there are, of course, several reasons for this, but one of them is that the low-hanging fruits are gone, and today's drug discovery pipelines tend to focus on diseases that are rather complex in their molecular nature, and hence require more sophisticated instrumentation to study these more challenging drug targets. Now, to this end, we have developed the new instrument Dianthus, that is the first nanotemper technologies instrument that works in 384 well plates. The instrument can be integrated into any lab automation and can measure a 384 well plate in 30 minutes. That makes it rather fast for a biophysical technology. Now, before I go into specific application examples, I would briefly describe how Dianthus works and how it conducts a measurement. Now, the technology that powers Dianthus is called temperature-related intensity change, or short TRIC. In TRIC, we usually label one of the binding partners. In general, that's the target protein with a fluorescent dye. And then we apply a brief laser-induced temperature change in the solution and then observe the variation in fluorescence intensity while we modify the temperature. You can see here in this illustration that typically upon an increase in temperature, the fluorescence of the fluorophore decreases. Now, interestingly, the change in fluorescence with temperature is strongly affected by the presence of ligands. Now, either the ligand binds in proximity to the fluorophore and can therefore affect its chemical microenvironment. On the other hand, a fluorophore, if distal from the binding site, can also be affected by conformational changes if a ligand binds. Now, this change in chemical microenvironment will change the susceptibility of the dye to change its fluorescent intensity when we modify the temperature. Now, you can see that illustrated on this slide. We have here the typical response that we measure. We usually measure a brief amount of steady-state fluorescence, then switch on the infrared laser to increase the temperature locally, briefly, and to a minute amount, and observe how the fluorophore fluorescence changes over time. We typically measure 5 seconds per well. And now you can see how the response is generally different if a ligand is bound or not. And that then results in a measurable signal amplitude that we can quantify. Now, if we titrate the ligand and then collect a dose response curve, we can plot the measured response against the ligand concentration and then extract quantitative values like the dissociation constant. Now that we know how the instrument works, let's get into specific application examples. And the first example that we want to have a look at is a fragment screen against a dimeric receptor protein. In this case, the protein is STING, which is short for stimulator of interferon genes. And STING plays an important role in innate immunity. It induces type 1 interferon production when cells are infected with intracellular pathogens, and it has been described as an interesting target to treat autoinflammatory diseases. Also, STING has been described as a promising immunotherapeutic target for different types of cancer. Now, in this case, we worked together with our partner Crelux, and they expressed a construct of STING for us, which consisted of the C-terminal domain, which contains the binding site for the nucleotides, and which was his-tagged. Why was it his-tagged? It is quite well known that it's difficult to modify the lysines of STING and keep the protein functional. That's why immobilization is usually not recommended. And we preferred an in-solution study where we labeled the protein via its his tag. Now, the longest part of the whole project was construct design and protein production, which took about 3 to 4 weeks, which was then followed by the assay development for the Dianthus screen. Usually we start assay development with data as shown here by a thermal shift assay to optimize the thermal stability of the target protein. And that took us around 1.5 weeks and 250 micrograms of protein. Now, the next step was then the fragment screen, where we screened 2,200 fragments in duplicate in single dose, which took us 8 hours of measurement time and 590 micrograms of protein. Then we selected a number of hits and titrated them to collect their dose responses and identify those fragments that would be most interesting to study further. Now, what this slide highlights is that for collecting this many data points, we needed less than 1 milligram of protein, and the measurement times were really fast. Now, looking at the resulting data, we see on the left-hand side the result of the single-dose screen, and each data point in the scatter plot corresponds to one ligand that was measured. And I want to point your attention towards the yellow dots, which correspond to the regularly measured positive control CGAMP. And you can see how CGAMP induces a reproducible and steady response over the entire screen. Now, on the right-hand side, you can see some data from the follow-up secondary screen where we titrated some of the fragments and interestingly found even ones with micromolar affinities that will be very interesting to study further. As a reference, I also plotted here the response obtained for CGAMP, which shows a nanomolar dissociation constant. Now, to sum up the results of the screen, we had here a very fast measurement speed with very low sample consumption, and we could mass independently detect binding of low-weight fragment binders. Also, we covered a broad affinity range that was only limited to the upper end by compound solubility. Now, next, I would like to show you an example for a protein-protein interaction where the target protein was a scaffolding protein of the postsynaptic density, a membrane-anchored protein. The ligand in this case was a peptide library, which is a sequence overlapping peptide library describing an interacting protein, in this case, a membrane receptor. Now, when we started with this project, we labeled the scaffolding protein by maleimide reaction and then titrated the peptides or added the peptides as ligands. And by this direct binding assay, we obtained this data. You can see it here on the right-hand side. Now what you might notice at first is that data points always appear in pairs. Now this is usually then a sign for a high assay reproducibility and quality, because these are the duplicates for each fragment that were individually pipetted. So the duplicates of the ligands showed a very nice response, reproducibility, but if we look closer, for example, at this hit, we see that the underlying raw data shows us a rather small signal response, meaning a signal amplitude. Now, we were not very happy with this, with these small signal amplitudes, and didn't want to go further with this assay. Now, luckily, Dianthus is a great instrument in terms of assay flexibility. And in this case, we just decided to use a labeled peptide as a tracer and first measured interaction with the protein. When we titrated the protein, we saw a very nice response with a large amplitude and a good signal to noise. Now, this prompted us to set up the assay in a way that we run a displacement assay, where we now titrate or add the peptide ligands to displace the labeled peptide from the binding site. And you can see here as an example 3 peptides that were titrated. And you can see how one peptide has a rather weak affinity and cannot displace the tracer, while 2 other peptides could in fact displace the tracer. Now, having such an assay with a good signal to noise and a large signal amplitude then made us very confident, and we decided with this assay to run an unpurified overlapping peptide library. And in fact, we could successfully confirm 2 binding sites that were previously described by other methods. Now, this data of this peptide library has been submitted for publication, and hence I cannot show it to you right now. But have a look out for the paper. We did this study together with our partners at the University of Würzburg, more precisely the lab of Hans Maric. So just have a look out for this paper. It will come out soon. Now, the final application example that I wanted to show is an investigation of multi-component interactions, and more specifically, PROTACs. Dianthus is a great tool to study PROTACs. Why is that? We have an in-solution technique, meaning we can very well describe binary KDs first and then describe a ternary KD in a second experiment. Now, how we proceed usually with PROTACs is we first measure the binary KDs by labeling, for example, the ligase and the target protein and titrating the PROTAC. That will yield these 2 KD values, which we call binary KDs, because they describe the binary interaction between 2 components. Now, by knowing these binary KDs, we can then preform a labeled complex of, for example, PROTAC and target, and then titrate the ubiquitin ligase as the third component, which will give us the ternary KD. And then from the ratio of binary and ternary KDs, we can calculate the alpha value, which gives us an idea about the cooperativity of this interaction. Now, to also give you an example, I want to show you this binding data for a PROTAC against a bromodomain protein. You can see here on the left where the bromodomain protein was labeled and the PROTAC was titrated, while here on the right-hand side, you see how the ubiquitin ligase was labeled and the PROTAC was titrated. And we have the KD values for these 2 interactions. Now, in the third experiment, a preformed complex of target protein, bromodomain, and PROTAC was formed, and then the ligase was titrated against that preformed complex. Now we obtained a different KD for the ligase because of cooperativity, and we can calculate the alpha value, which in this case was 78. Now, I think this data speaks for itself. It has a very nice signal to noise and shows how Dianthus is very well suitable for studying ternary complexes, also, maybe especially in the context of PROTACs. Now, to sum it all up, these are the 3 take-home messages, I think, from these application examples. We have seen mass-independent detection of interactions within a broad affinity range for a fragment library against sting. Also, we have seen that Dianthus offers a high degree of assay flexibility with your assay design with a peptide example. And finally, we have seen how Dianthus enables you to perform in-solution studies of complex oligomeric targets such as PROTACs. With this, I would like to close this webinar. Feel free to contact us with any question. They are always appreciated. And I wish you a nice day. Thank you very much.